Hello, and a warm welcome again to this lesson today. Welcome to Nestor's Bioacademy. And today, the main focus of this lesson today is to describe water relations in plants and animals. This is the second lesson on osmosis. In case you missed the previous one, you can access the previous lesson on osmosis in the video. Go through it and then get back to this second lesson on osmosis. So the main objectives of this lesson today is to describe what happens to plant cells when you place them in solutions of different concentrations, and also describe what happens to a red blood cell when you place it in solutions of different concentrations. Remember previously in our earlier lesson on osmosis, we defined and reviewed and explained what osmosis is. We looked at the features of osmosis, and we also described the terms we always use to compare concentrations of different solutions. The terms hypotonic, hypertonic, and isotonic were well described. Remember, we defined osmosis as the movement of water molecules from a dilute solution to a concentrated solution across a semi-permeable membrane, or we said you could talk of it as a movement of water molecules from where the water molecules are highly concentrated to where the water molecules are less concentrated across a semi-permeable membrane. So in a nutshell, we said osmosis is simply diffusion of water because the water molecules are moving from a region where they're highly concentrated to where they are lowly concentrated. Then we looked at the various terms we use to describe concentrations of solutions: hypotonic, hypertonic, and isotonic solutions. So remember we said a hypotonic solution is a solution with a less solid concentration compared to another solution of reference. So ideally, hypotonic describes a solution which is dilute compared to another reference solution. But a hypertonic solution was a solution that has got a high solid concentration compared to another reference solution. So this is essentially a concentrated solution. But when two solutions are having uh, uh, identical concentrations of solute and solvent, then we say that the two solutions are isotonic to each other. So the key questions we are going to attempt to answer today are the following. What happens when a plant cell is placed in solutions of different concentrations? And what happens when an animal cell, like a red blood cell, is placed in solutions of different concentrations as well? So to start with the plant cells, what will happen to a plant cell when it is placed in a solution of, in either a hypertonic solution, isotonic solution, or a hypotonic solution? What is it that is going to happen? Remember, the key thing to always note is that water molecules will always move from a dilute solution to concentrated solution. And if you can recall from our previous learning when we're looking at the topic of the cell, remember we learned that the cell is, the plant cell usually has a large centrally placed subvacuole, and this subvacuole usually contains it contains a fluid which has got dissolved salts and sugars, which, if you recall, we were referring to as the cell sap. So this means we are having a solution within a subvacuole, which you can compare its concentration to the solution in which the plant cell has been placed. So when we talk of these terms, hypertonic solution or hypotonic solutions, these will be comparative terms to the cell sap of the plant cell we are talking about here. So meaning when you're talking about a hypertonic solution, it means the solution is hypertonic to the cell sap. If you're saying it is hypotonic solution, it means it is hypotonic to the cell sap. Remember, we say hypotonic or hypertonic to the cell sap. We are not going to say that the solution is hypertonic to the cell. Because these terms, hypertonic, hypotonic, isotonic, we use them to compare concentrations of solutions. But a plant cell is not a solution. The solution is the cell sap, which we find inside the plant cell. So if we were to place a plant cell in a hypertonic solution, this will mean that the solution is having a higher solid concentration compared to the cell sap, and therefore it will mean the cell sap is dilute compared to the solution in which it is placed. So what is going to happen? The plant cell is going to lose water by osmosis, and as it loses water by osmosis, we expect the sap vacuole to decrease in volume, and as it decreases in the volume, the cell membrane will be detaching from the cell wall, like you can see in this scenario. So what eventually happens is that the cell becomes plasmolyzed. The cell tends to shrink in size, and the cell membrane is going to detach itself from the cell wall. If we could look at this illustration further, for greater emphasis, this is the cell wall of the plant. We always have the cell membrane firmly pressed against the cell wall. And of course, we're having the sap vacuole where we are saying we're having the cell sap, which contains salts and sugars. So it is basically a solution. So if we take the plant cell and we place it in a hypertonic environment, the cell sap is going to be dilute compared to the solution in which it is placed. So due to osmosis, water molecules will move. And as water molecules move from the plant cell, we expect to see the cell membrane. Now this is the cell membrane. So the cell membrane is going to detach itself from the cell wall. So in this state, the cell is said to be plasmolyzed. And in this state, you can also say, describe the cell as being flabby. And this means the cell is less rigid or less firm. And so the process by which a plant cell loses water when placed in a hypertonic solution to shrink in size and have its cell membrane detaching from the cell wall, we call this process plasmolysis. And when this happens, the cell becomes relatively flabby or less firm compared to the normal plant cell. So when a plant cell is placed in a hypertonic solution, the cell sap will be hypotonic to the solution. So the plant cell will lose water by osmosis to become plasmolyzed. But in a hypotonic environment, this means we now place the plant cell in a hypotonic environment, meaning if the solution is now dilute compared to the cell sap, so the cell will instead gain water by osmosis, and as it gains water by osmosis, it stretches and then it becomes turgid, or it becomes very firm. It becomes very firm. We will later on look at the significance of this turgidity or firmness, or the rigid state that the plant cells obtain once they gain water by osmosis. So it's a common, you can commonly be asked to describe what happens to a plant cell when placed in a hypertonic solution, or describe what happens to a plant cell when it is placed in a hypotonic solution. Or we can sometimes even ask you to make illustrations of a plant cell when it is placed in a hypertonic solution. So the knowledge of the behavior of the cell, the detachment of the cell membrane when the cell becomes from the cell wall, when the cell becomes plasmolyzed is very key there. Next, we also need to look at what happens to a red blood cell when it is placed in solutions of different concentrations. So what is going to happen to a plant cell, to a red blood cell, when you place it in a hypertonic solution? Even though the red blood cell is not having the centrally placed subvacuole, so we don't have the cells up in this case. But remember, we also have the cytoplasm, or basically the cell contents, which is also which is going to be the solution of reference, comparing it to the solution in which the red blood cell is placed. So when you talk about a hypertonic solution, it means the solution is more concentrated compared to the red blood cell cytoplasm or the red blood cell contents. So this will mean that the solution will be hypertonic to the red blood cell contents. So the red blood cell is going to lose water by osmosis. When it loses water by osmosis, you expect the red blood cell to shrink and become crenated. The illustration here represents the crenated nature of the red blood cells. And you can see the red blood cells end up losing their biconcave shape. And this biconcave shape, remember, is very important in ensuring that the red blood cell is more efficient in transportation of oxygen and carbon dioxide in the body. So this means when a red blood cell is to be in a hypertonic environment, its functionality is going to be significantly affected. So this process by which a red blood cell loses water by osmosis to a hypertonic solution to shrink and become shriveled is what we refer to as crenation. How about a red blood cell in a hypotonic environment or a hypotonic solution? So in this case, it means the solution is more dilute compared to the red blood cell contents. And remember the direction of osmosis. Water molecules will always move from the dilute solution to the concentrated solution. So if the solution is hypotonic to the red blood cell, we expect the red blood cell to gain water by osmosis, then it will swell and burst, or it will undergo lysis. And this happens because unlike the plant cell, which was not undergoing any sort of bursting, the red blood cell does not have a rigid cellulose cell wall that can prevent that. We are going to explain this in a short while. So in a hypotonic environment, you expect the red blood cell to gain water by osmosis, swell, and eventually burst. We refer to this process as hemolysis. Again, you don't expect a red blood cell that has undergone lysis to have any functionality. But in an isotonic environment, the red blood cell will retain its biconcave shape, and that is going to be important in ensuring that the red blood cell carries out its function in an effective way. And this therefore tells you that we always expect the concentration of the red blood cell cytoplasm to be nearly isotonic to the concentration of the plasma. Remember, plasma is the fluid in which the red blood cells are always suspended. So in a nutshell, this illustration is summarizing what happens to the plant and red blood cells when placed in solutions of different concentrations. When placed in a hypotonic environment, the red blood cell is going to undergo hemolysis. They will take in water by osmosis, swell, and eventually burst. But when placed in a hypotonic solution, the plant cell will gain water by osmosis, stretch, and become rigid or become very firm. When placed in isotonic solutions, both the red blood cell and the plant cell, they will retain their normal shapes. In an isotonic environment, the plant cell will have a normal appearance. It will not be flabby or very much less rigid. It is also not going to be very turgid. So that condition is referred to as a flaccid condition. In a hypertonic environment, we expect the red blood cell to lose water by osmosis, and when this happens, our red blood cells are going to be shriveled, or they're going to be crenated. We refer to this process as crenation. And then when we take a plant cell and we place it in a hypertonic environment, the plant cell is going to lose water to the solution, to the hypertonic solution through osmosis. And when this happens, we expect the subvacuole contents to shrink. And when this happens, the cell membrane detaches itself from the walls of, from the cell wall. And this, in this state, the cell is described as plasmolyzed. It becomes less firm. Now, the turgidity, when a plant cell becomes turgid, it becomes very important in ensuring that the plants remain upright. And this upright nature is very important because it enables the plants to expose their leaves well to the environment. So sunlight trapping is more efficient for the process of photosynthesis. If the plant had flowers, it will expose its flowers better to pollinators. If the plant cell, if the plant had fruits, they'll be well exposed to the dispersal agents. But if a plant cell, or if the plant cells were to lose a lot of water, it will mean the cells will become less rigid, they'll become plasmolyzed. When the plant cells become plasmolyzed and less rigid, the plants will lose their support. They will not be able to, the plant tissues will not be able to stand erect and in an upright way. So the plant will undergo what we call wilting. And in this state, the plant is not able to expose its leaves well to trap maximum sunlight for photosynthesis. However, there is an advantage in this in the sense that when the plant cells become plasmolyzed and less rigid and less firm, mostly we expect the leaves to tend to roll, and when the leaves tend to roll, they reduce the surface area exposed to sunlight, and this enables the plants to reduce the amount of water they lose through transpiration. So wilting is a phenomenon that occurs when a plant loses more water than it is absorbing. So the plant cells become plasmolyzed and they lose that turgidity. So the leaves and the shoots of the plant, they tend to droop. They tend to droop. Remember, we can liken this scenario to a balloon. If I was to have a deflated balloon and hold it upright at the tips of my fingers, the balloon will just droop. It won't be able to stand upright. But when I now inflate it, if I blow air through it, by having more air now, it's able to stand firm if I was to hold it at the tips of my fingers. Over to other observations, you must have realized that red blood cells, they will burst in a hypotonic environment, but the plant cells do not burst in a hypotonic environment. You must have also realized that the plant cells, they maintain their shape in different solutions, while the red blood cells were easily losing shape. They were either becoming crenated or they were becoming lysed. So that begs the next question. Why are the plant cells not losing their shape compared to the red blood cells? And why were the red blood cells bursting while the plant cells are not bursting when they are placed in a hypotonic environment? The answer lies in one structure present in the plant cells and absent in the animal cells, as we learned in the previous topic of the cell. Now, the red blood cells will always burst because they lack the cell wall. The cell wall is always made up of a rigid cellulose that can resist the pressure exerted on the cell wall, on the cell membrane as water enters the cell. So remember, the plant cells have it, but the red blood cells do not have it. The red blood cells are only having the cell membrane, which is not able to resist the pressure as water enters the cell by osmosis. So usually when water enters the cell by osmosis, when the plant cell is placed in a hypotonic environment, we expect an increase in the subvacuole. So as water enters the subvacuole, the contents of the subvacuole will be increasing. And as this happens, the subvacuole will be exerting some pressure, some outward pressure against the cytoplasm, which again exerts this pressure on the cell wall. So this pressure that is exerted on the cell wall by the cytoplasm due to water intake as the subvacuole expands, we refer to this one as the Tagger pressure. So the Tagger pressure is exerted by the subvacuole on the cell, on the cytoplasm, and against the cell wall. In return, the cell wall being made up of a rigid cellulose will exert some opposite pressure, which is equal to the Tagger pressure. And this is the pressure that prevents the plant cell from bursting. So the wall pressure is the opposite pressure to the Tagger pressure that is exerted by the rigid cellulose cell wall, and it performs a critical role in preventing the plant cells from bursting, like what we are seeing with the red blood cell, which is bursting because it's lacking the cellulose cell wall. Now, remember we said when a plant cell is placed in a hypertonic environment, the plant cell was going to lose water by osmosis, and then the cell membrane detaches itself from the cell wall, making the plant cells to be plasmolyzed. Now the question is, are we able to restore a plasmolyzed cell back to the normal cell? And the answer is yes. If we are to take a plasmolyzed cell and we place it in either distilled water or a hypotonic solution, it will simply absorb water by osmosis to become turgid or normal once more. We refer to this process as deplasmolysis. It is simply a process of reversing plasmolysis, and we do this by placing the plasmolyzed cell either in distilled water or in a hypotonic solution, and that is going to reverse the process of plasmolysis. And then we also have the factors affecting the rate of osmosis. Now, do you recall the factors affecting the rate of diffusion? If you do recall the factors affecting the rate of diffusion, then there are similarly the same factors which affect the rate of osmosis, because remember, as we have said earlier, osmosis is basically a special type of diffusion. Both osmosis and diffusion are essentially passive processes. Both of them are processes which do not require energy. All that they require is a difference in concentration of molecules between any two regions. So the factors affecting the rate of diffusion are the same factors that will affect the rate of osmosis. Now, a sample response is here. I want us to look at this question carefully. Describe what happens to a red blood cell in a hypotonic solution for 3 marks. If you are to respond to this question, and I want us to look at these two possible responses, I have response A and I have response B. Response A, red blood cell contents will be hypertonic to the solution. That is correct. So the red blood cell will gain water by osmosis, mention of the process very key, and then it will swell and burst due to lack of a rigid cellulose cell wall. And this answers the 3rd mark, because the question is explain what would happen. So meaning there's need for the candidate to tell us what will happen and then a reason for it. So this response scores well for this question. But let's look at this response, response B. Response B, the student says the red blood cell contents will be hypertonic to the solution. At this point is correct. But then let's read the next question. The red blood cell will gain, the red blood cell gains the solution by osmosis. We lose from here. And kwa sababu, the student says the red blood cell gains the solution. You must always desist from referring to osmosis as gaining or losing a solution, because osmosis is strictly movement of water or solvent molecules. So a lot of care must always be taken when dealing with questions like this. In this first point, red blood cell contents will be hypertonic to the solution. It can still be the solution will be hypotonic to the red blood cell contents. This is simply an aspect of comparison. We are comparing what is the concentrations of the red blood cell contents to that of the solution in which the red blood cell is placed. And then we are mentioning the process. The water molecules are going to move through which process. And finally, we are relating it back to the observation. So mostly for questions of osmosis, it's always ideal to track your responses to ensure you are comparing the concentrations of the solutions, you are mentioning the process, and you're relating it to the observation that is going to be made in that regard. And again, remember, for the comparison can be made in two ways. I can say Tom is taller than John. That means I can also say John is shorter than Tom. So either way, the aspect of comparison needs to come out effectively. And again, you must always desist from saying the red blood cell would gain the solution or would lose the solution, because that will not be osmosis. You can track if you can respond to these questions. Why should the concentration of blood plasma and that of the red blood cell cytoplasm be kept at nearly the same concentration? A plant cell does not burst when placed in distilled water. What explanation can you give for that? And are you able to make an illustration of a totally plasmolized cell? And if you can recall from the previous video, what makes osmosis a special form of diffusion? Away from that, it's important that we also look at the critical roles that osmosis plays in living things. And osmosis is a very important process in absorption of water from the soil by the roots. Remember, if you can recall from the previous topic, when we're looking at the specialized cells, we said the roots usually have the root hair cells. And the root hair cells, usually they contain, if you recall, they have an elongated projection that way. And inside, we are having a sap vacuole that we learned was greatly concentrated in salts and sugars. And in the surrounding soil, we have soil water. So the water here in the surrounding soil will always be dilute compared to the sap vacuole there. So we always have water molecules moving into the root hair cell sap by osmosis. And once the water moves into the root hair cell sap, it is then moved into the other plant tissues through the process of osmosis. And other forces we'll be learning later on in Form 2. So apart from absorption of water from the soil by roots, movement of water from cell to cell in living organisms, osmoregulation in animals and other unicellular organisms, support in herbaceous plants by maintaining turgidity, and also opening and closing of stomata. Of course, there are other roles you can also mention, like reabsorption of water at the kidney tubules. And of course, that one is contributing to osmoregulation. We also have absorption of water from the alimentary canal in our bodies as animals. So this makes osmosis a very critical process in supporting life in our bodies. And so the key takeaway for this video, there's need for you to be able to describe what happens when a plant cell is placed in a hypertonic solution, hypotonic solution, and in an isotonic solution. What happens when you also take a red blood cell and you place it in these solutions of different concentrations? And we have also mentioned a few terms which you need to review and ensure you are able to describe them in the context in which we have described them today. Crenation, hemolysis, plasmolysis, deplasmolysis, flaccidity, and turgidity. So that brings us to the end of this session for now. Till the next video, see you.